Hello everyone, my name is Rama. I'm a fourth year PhD candidate at UConn Health and I work in the labs of Dr. Philip Smith at Center on Aging and Dr. Steve Crocker at the Department of Neuroscience. And my talk is about the regulation of bladder function by central nervous system myelin and its implications for multiple sclerosis. The circuit of micturition includes the brain, the brainstem, the spinal cord, the peripheral neurons, and the bladder. And the nervous control of bladder function involves the central nervous system neurons whose axons are wrapped by myelin that ensures faster flow of electrical signals by acting as an insulation, and it also provides trophic support to the neuron. So without myelin, nerve signal transmission will be slower and sometimes not effective, disrupting communication and coordination, leading to loss of function. This is commonly seen in diseases such as multiple sclerosis. Multiple sclerosis, or MS, is an autoimmune disease characterized by inflammation caused by autoimmune attack by inflammatory cells that invade the central nervous system and destroy the myelin sheath, causing demyelination, thereby slowing the neuronal signals. And chronic demyelination eventually leads to axonal loss since there is loss of trophic support, and all this causes deterioration and eventually loss of function in different regions of the periphery based on the extent and location of the inflammatory demyelinating lesions in the central nervous system. So different MS patients have different functional disturbances based on where they have lesions in their central nervous system. MS is a debilitating disease that affects over 2.3 million people worldwide, and women are 3 times more affected than men. And of note, over 80% of MS patients have urinary symptoms as one of their earliest symptoms, which includes hesitancy, urgency, frequency, and incontinence. And medically and psychologically, these symptoms have a pretty dramatic impact on their quality of life. The exact mechanism by which MS causes these bladder symptoms is not completely known, so the goal of our research is to determine the link between what happens in MS as a disease and the effects seen in bladder function. Canonically, it is thought that lesions in the spinal cord cause MS-associated bladder dysfunction. However, there are other regions such as the brain where MS lesions could potentially develop, causing bladder dysfunction. The role or impact of myelin in the brain or the cortex towards regulation of bladder function has never been established before, so we asked, where in the nervous system is the bladder controlled by myelin? To address this knowledge gap more specifically, we hypothesized that the loss of myelin in the brain is sufficient to cause bladder dysfunction, and that conversely, remyelination can restore control. To test our hypothesis, we used the Cuprizone model in mice. Cuprizone intoxication is a non-immune model of central nervous system demyelination. In this model, young mice fed Cuprizone diet undergo complete demyelination in 4 weeks. So these electron microscopy images show a cross-section of several axons in the brain of these mice. You can see how 4 weeks of Cuprizone treatment, which is the right panel, denotes axons that are gray of myelin, which is the black outline. And after 4 weeks of demyelination, switching the mice to normal diet restores their myelin and causes robust remyelination, or continuing them on Cuprizone diet for 8 more weeks takes them to a point where their spontaneous remyelination potential is impaired and demyelination sustains. So using the Cuprizone model, we assigned 4 different treatment groups: the untreated, the 4-week Cuprizone treated, the 8-week Cuprizone treated, and 4 weeks of Cuprizone treatment followed by 8 weeks of recovery from Cuprizone allowing remyelination. The Cuprizone model is a classic model that develops profound demyelination in the brain with no known demyelination in the spinal cord. So we used a very interesting strategy to tease apart the effects of brain demyelination on bladder function. To evaluate changes in the control of bladder function, we performed voiding spot assay in these 4 groups of mice. After that, we blocked the cortical or brain inputs using urethane and then evaluated the involuntary reflex to bladder filling using urethane anesthetisostometry. In both these experiments, we evaluated the frequency of micturition and the voided volume. In the voiding spot assay, mice were individually placed in cages lined with filter paper for 4 hours with food but no water. And after 4 hours, the filter papers were UV imaged and analyzed. So the voiding spot assay revealed significant changes in urinary behavior of Cuprizone-treated mice. With Cuprizone treatment, the number of urine spots increased as seen at 4 weeks of treatment and 8 weeks of treatment. And most importantly, when we computed the total amount of urine based on total area of spots, it was the same. shows that all these treatment groups expel the same amount of urine in the given 4 hours. Therefore, instead of having fewer longer micturition cycles, which is healthy, the Cuprizone treated mice with demyelination had more frequent, shorter cycles of micturition. Interestingly, the group that underwent recovery and remyelination had bladder function similar to control unlesioned animals, suggesting that Cuprizone recovery and remyelination rescues normal urinary behavior. Therefore, we have determined that loss of bladder function in mice is coincident with loss of myelination in the brain, and the bladder function control is returned when myelin is regenerated. Next, we performed cystometry on these mice under urethane, and our results show that the frequency of micturition and the voided volume as a result of involuntary reflex did not change due to Cuprizone treatment, but this is expected from a model that has only established brain demyelination that affects conscious cortical control. So as a summary, Cuprizone treated mice with cortical demyelination and no spinal demyelination display bladder dysfunction, as shown in VSAs, with increased micturition frequency and reduced volume per micturition. However, when the cortical inputs are blocked using urethane and the bladder spinal reflex is evaluated, the Cuprizone treated mice behave similar to control mice as seen using cystometry. So putting this together, our novel finding is that the loss of myelin in the cortex causes increased urination frequency, and this is suggestive that urge incontinence, especially in MS patients, could also be a sign of cortical demyelination, and clinically, to test MS-associated bladder symptoms, remyelination therapeutics could be a better choice than oral bladder-specific drugs, which is currently the mainstay therapy for these patients. Thank you so much for your time and attention.